Okay, this lecture is to talk to you about vaccines, antivirals, and kind of the tools we have to deal with or prevent virus infections. So, vaccines are preventative. And this is the most recent. Well, actually, I think there's another one that's more recent. Okay, start. Okay, this lecture is to talk to you about vaccines and antivirals, and basically some of the tools we have to either prevent or deal with viral infections. So vaccines are our primary method of prevention. And this chart is showing you the vaccine schedule for those 0 to 18 years of age. So you can see you start with hepatitis B, and then you get rotavirus and diphtheria, tetanus, pertussis. We'll talk about some of these. Haemophilus influenza B, which is actually a bacteria, pneumococcal, polio virus. You continue to get boosters for those. You add influenza, measles, mumps, rubella. As you get older, meningococcal to prevent meningitis, hepatitis A, varicella, which is chickenpox. And then as you get into your teens, human papillomavirus, and every 10 years you got to keep getting your tetanus update. Here's the vaccine for you all, 19 and over. So again, your every 10-year tetanus booster, influenza, papillomavirus, human papillomavirus, if you didn't have those. Some people are getting measles, mumps, rubella, MMR, depending on situations. And then, again, a lot of these say depending on indication. And then as we get older, here we go. Zoster is the shingles vaccine. So after you're 60. And we'll talk about some of these viral vaccines. So this is a mix of vaccines to prevent viral infections and vaccines to prevent bacterial infections. I think this is an interesting chart. Just the big difference between when I was a kid, how many vaccines we got, to what people are getting, children are getting now. And it's a lot. It's a lot of vaccinations. And as you know, there is a lot of controversy, excuse me, with vaccinations. And I'll touch on that a little bit. I questioned my children's pediatrician when they were little, like, why do they need a chickenpox vaccine? Why do they need all these vaccines? And I talked, of course, the pediatrician recommended it for safety and protecting them from preventable diseases. And I talked to people who were in research for immunology, and they were all very positive. So I am definitely a pro vaccine person. So where did we get vaccines from? So back in the late 1700s, there was this observation, and one of the big, what do I want to call it, diseases, deadly diseases back then was smallpox, which is caused by a virus. And the observation was, one, if you ever got smallpox and survived, you never got it again. And two, milkmaids who would get pox like blisters from milking the cows never got smallpox. So some of the first vaccinations that were tried was that they would actually take, so smallpox, this is your skin, smallpox will form this pustule. And I have some pictures of smallpox later in the talk. Right? So this has got pus in it, and it's shedding virus, and it's your reaction to the virus. Unfortunately, smallpox can cause death. But what they tried is, based on those observations, that if you had smallpox once, you never got it again, they would take a little bit of this pustule, and then, and this is my great drawing, here's a person, inoculate them with it. So they were inoculating with live smallpox, and seeing if they could kind of give it to this second person and prevent them from getting it again. It was a 20% death rate. So 1 out of 5 people who got the smallpox vaccine back then from the live virus died. So that wasn't really a good way to prevent smallpox. So a scientist named Edward Jenner came up with the idea, based on the second observation, that milkmaids never seem to get smallpox. So this little kiddo here, in 1796, this 8-year-old boy, who was the son of Jenner's gardener, was inoculated with cowpox. So what they're trying to depict in this picture is, here's the milkmaid. She had a pox pustule from the cow pox virus, and so they scraped a little of this, and then they scraped it on the boy's arm to inoculate him. This is what I find amazing. Not only did someone kind of volunteer their son for this, but then Edward Jenner, and I can't find the timeline, challenged the boy with live smallpox virus. So he actually, to make sure this vaccine was going to work, gave the boy smallpox. Right? The boy survived. He did a second challenge later. The boy survived. And this was our first true vaccine where you're not using directly the live infectious agent, but some variation of it. So what we use now is a virus called vaccinia virus, and vaca comes from cow. It's not the one that causes cowpox. It's not the one that causes smallpox, but it's a related virus. And if you were born before about 1978, maybe it's the 70s now, my brain is getting old, or in the military, you may have a scar on your arm. I have one from where they gave the vaccinia inoculation. It indeed forms this little pustule. You recover and you get a nice scar. So, you know you're vaccinated. In the late 1970s, I think it was 78, we stopped here in the States giving the vaccinia virus smallpox vaccine, and as we'll talk about, because smallpox was eradicated, so wiped out. There was no more smallpox transmission. So this is where vaccines come from. The goal of the vaccine is long-term protection. So the big goal here is really that you're practicing, well, let's back up. The goal is to stimulate your immune system. Your immune system is what either prevents or fights off infections, whether they're viral, protozoan, helminth, bacterial, any of those microbes. So the idea of a vaccine is we're going to give you some, not the strong disease-causing, and we'll talk about different types of vaccines. Let your body practice, and then if you are exposed to the real, in this case virus, you're going to mount a much faster immune response. Which should prevent or at least minimize disease. Prevention is the key. Sometimes we actually have a little bit of disease infection that we don't even know because it's so minimal. We'll talk about, I think the next slide. Yeah, let's talk about it right now. So the goal of vaccines is to stimulate the part of your immune system called the adaptive immune response. And this is really where the memory comes in. So you want to be able to create antibodies, but you want it with memory B cells. So what's happening during a vaccination is that antibody production is stimulated, and those antibodies that work strongly against the vaccine will become activated B cells that will be secreting antibodies, and then they will become memory B cells. So your body is ready with these antibodies, and every time that B cell is activated, it sees that specific foreign invader. It can make a whole bunch of antibodies and hopefully wipe out the infection. The other part of your memory is the cell-mediated, right? So T cells. And T cells are great because they also recognize very specific receptors, and they can attack infected cells. So we have lots of T cells floating around, and when they become activated due to this practice with the vaccine, they become memory. So you've got antibodies that are trying to prevent the infection and T cells that are attacking infected cells. It takes a couple weeks to get this for a vaccine to work. So let me go back. So this is about 14 days at least that you want to have a vaccination and get this practice before you have or exposed to the real infection. So again, vaccines are preventative, and you need to take them early enough. So that's why they try to get you to get your flu shot early in September, October, before the real flu virus is heavily circulating, so that your body has time to get that antibody and T cell response up and going. Something that's really been talked about a lot is the idea of herd immunity. The idea is that you probably can't get 100% immunization, and herd immunity is down here. And there are different reasons for that, and again, we'll talk about this a little bit as we talk about the different types of vaccinations, but there are some people who cannot get vaccinated. Okay? They're immune compromised, their body can't deal with that. So not everybody can get vaccinated. So this is our goal of herd immunity. And the idea behind it is that you're breaking the line of transmission. Okay? So if you have enough people that are vaccinated, those that aren't, and they become sick, the virus or the bacteria doesn't have anywhere to go, because when it's passed on to these vaccinated people, their bodies fight it off and they stop the chain of transmission. You have to have enough people vaccinated for this to work. Okay. So yes, the vaccinated people should be protected regardless, but it's these not immunized but still healthy people that we worry about, the people who cannot get a vaccination. So let's talk about what are some of the types of vaccines. Let me give you a couple of definitions, and then we will go through these more in more detail. We're going to talk about vaccines for viruses. The same types of vaccines can be made for bacteria. These two, attenuated and inactivated, are very common for viruses. So attenuated means a weakened but infectious virus. Okay, so that's why we also call it live. So you'll hear attenuated, you'll hear live. Live doesn't mean we're giving you the virulent disease-causing one. We're giving you a weakened version. And most of the time, this weakened version is made by growing the virus, so having it go through replication cycles in a less than ideal condition. And this less than ideal condition forces the virus to create some mutations, to adapt, to make more virions. Then we take this weakened virus and put it into humans, and the virus doesn't cause disease because it's now not human, what we call human adapted. So it will go in, it will do some replication, but it's not likely to cause disease. It's probably more slow growing due to the mutations, and so your body has a chance to respond to it to prevent infections from the true virulent virus. Inactivated or killed means it is the virus that is no longer infectious. Okay. So we can do this a couple different ways. We can grow up a whole bunch of virus in tissue culture, isolate the virus, treat it with chemicals so that there's no, say, uncoating. We might treat it with chemicals so that it has a non-functional genome. So a lot of times they'll do cross-linking between the nucleotides or between proteins in the nucleotides using heat, and so the genome can't replicate, can't be expressed in the cells. So inactivated, killed, your body still sees the outside of the virus like it was a real one, but it can't cause any kind of infection. Subunit vaccine, we have some for viruses, we'll talk about. This is really more common for bacterial, but as you can imagine, a subunit means part of the virus outside. So if we go back to this picture about the goals of the vaccines, I want you to understand that these antibodies are recognizing something on the outside of the antigen. And antigen just means something that antibodies bind to. So this could be a bacterial cell, this could be a virus, this could be part of the outside of a virus. Okay. So a subunit vaccine, we just take part of the virus outside and use that to stimulate the immune system. So let's talk about, some of the pros and cons of attenuated and inactivated vaccines. The pros of attenuated is this is best at stimulating the immune system. There we go. Because this works like a weak infection, you're getting a full immune system response, B cells, T cells, all of that going, and it makes a very long-lasting response, which means you usually have to have decreased number of doses because it's long-lasting and it's such a great stimulator of your immune system. So this is, again, a weak virus that's live. It does an infection. It just shouldn't cause disease. The negative is that keyword shouldn't. So there is potential to cause disease. And I'll talk about this when I talk about the polio vaccine in just a few slides. Every once in a while, you get a revertant. So remember, the attenuated vaccine is made by growing the virus in less than ideal conditions. It accumulates mutations so that it doesn't replicate very well in humans. Those viruses can, those attenuated viruses can still replicate, or they do still replicate, and potentially revert back to a virulent state. Very, very rare, but it is potential. The other negative is it cannot be given to people that are immunocompromised. So, older people, very young people, people with organ transplants, people with cancer undergoing chemotherapy. There are different people that absolutely cannot get a vaccination, especially with an attenuated one, because it's replicating. It's going to, and they don't have this strong immune system, it's actually going to cause not necessarily disease like polio, but it can cause overstimulation of what immune system they have, and allow for secondary infections. So we just don't give attenuated vaccinations to immunocompromised people. Another negative is it requires cold storage. Just like you put your food in the refrigerator to keep it fresh, you have to keep these weakened live viruses fresh, and so you have to have cold storage, which may not seem like a huge deal here, but in other countries, they may not have electricity, they may not have access to refrigeration, and so trying to get vaccines, attenuated vaccines, to these people is a huge challenge. The other potential negative is that let me write this differently. Vaccinated people can transmit live virus, okay, which can be great in the sense that it can help with herd immunity by you vaccinating this person, and they might transmit the virus to this healthy person and they become vaccinated as well. But, oops, I'm going the wrong way. You can imagine that this is an issue for immunocompromised people, or again, elderly or young people. So that's a challenge with attenuated. Inactivated, which are killed, the biggest positive is it cannot cause disease. Why this does this? Okay, there's absolutely no way it can cause disease because it has been inactivated. It's very stable. So it does not cause, or I'm sorry, looking at the thing above it. Very stable, so you don't need refrigeration. So this is great. We can get this vaccine to more people. Sorry. Those of you who know me know I have a lot of computer issues, and I don't know why. Some of the negatives, as you can imagine, the immune response is not as strong. Okay, which means you're going to require boosters or multiple doses. Another negative is that inactivated must be injected. Although we're all really used to our vaccine shots in the olden days, and I'll talk to you about this for polio, is that they would put the live attenuated polio virus vaccine on a sugar cube and give it to the kids. So you can imagine how much stress this reduces without having to get a shot. And the other negative is you don't get as full range of immunity. Not full immunity. So this is very true in that it doesn't stimulate the mucosal immunity. You also sometimes don't get activated T cells. So remember, T cells, as I showed you here, are going to work against infected cells. So if your cells are not being infected because it's an inactivated virus, you're not going to get all of that, both antibody and T cell immunity. Now remember, and this will become important in the next slide too, some viruses are taken in. We talked about the life cycle or replication cycle of viruses. Some viruses are taken in by receptor-mediated endocytosis. And in that case, you definitely would get the T-cell immune response because the inactivated virus has been taken into the cell and then the antigens presented on the outside. So it just kind of depends on that specific virus. Subunit and virus-like particle vaccines. So these are both just outside parts. So a virus-like particle is similar to that inactivated, but there's actually no genome. So both of these are very safe. Just outside, no possibility for disease. There's no genetic material. All the components are not there. So a recombinant subunit means it's just part of the outside. This is showing like a spike on one of the outsides of the virus. Virus-like particles, if you remember when I was talking about assembly of viruses, you can put the capsid pieces, into a test tube and they will self-assemble into these little empty capsid shells. This does not work for enveloped viruses. So only naked viruses. Because these viruses, they're just the protein coat. You can't get them to bud. This can be good for both B and T cell immune response because a lot of naked viruses are taken into the cell by receptor-mediated endocytosis, as I said previously, and so they do kind of look like a virus infection, and you can get a T cell response. This one, only B cell response. So you're only getting antibodies to this outside part. So just the outside, no possibility for disease, non-infectious, is kind of what I just said. Just looking at my notes here. The challenge, especially with subunit vaccines, and we see more subunit vaccines, like I said, with bacteria, is to find that right part to make something that we call a neutralizing antibody. That didn't change. So that is our goal. Make a neutralizing antibody, one that is going to prevent infection. So if we look back on this slide here, we want an antibody that is going to neutralize this antigen and it can't ever even infect. That's ideal. And that's hard to figure out the right subunit. So the first paper we're going to look at, influenza antibody paper, is about trying to create a subunit vaccine that makes these neutralizing antibodies. So those are the main types of vaccines for viruses, and I've listed some of the common virus vaccines here. I did not include the bacterial ones. You can see measles, mumps, rubella, 3 attenuated viruses, polio. We're using an inactivated. I'll talk about these 2 vaccines in a minute. Influenza, primarily inactivated. A few years back, they used an weakened attenuated nasal mist, which I got, because I hate shots. And how that attenuated one worked is they caused mutations in the influenza that would only allow it to replicate in the cooler nose space. And if you breathed it in and got it in deeper into your lungs where the temperature increases, the virus could not replicate, and so it couldn't cause disease. HPV is really struggling with virus-like particles. They are taking, and there's even a HPV-9, I think now, that they're actually taking a combination of the different capsids from different strains of human papillomavirus and allowing them to assemble into these kind of recombinant virus-like particles and making a really great vaccine to prevent human papillomavirus, which is can be a sexually transmitted sexually transmitted cancer. Yellow fever, something we don't talk about in this country, but it's one of the oldest, most safe vaccines. Rabies, we normally don't get a vaccine against the rabies virus. We have something called post-exposure vaccine, which is an inactivated vaccine, or people who are animal handlers or spelunkers, people who go into caves, can get a rabies vaccine, but it's not something that's commonly given to people. So let's talk a little bit about some vaccines you probably have heard of. So poliovirus, something we don't really worry about in this country, but there was 2 different vaccines created. So the Sabin vaccine, is the attenuated. So it's the live virus. Okay. And so therefore, people shed live virus. Okay. So poliovirus is transmitted through the fecal-oral route. So you get polio from ingesting contaminated water or food. And the virus gets there by people shedding it in their feces. Polio, as I mentioned, is very stable in the environment, like in the soil, months. In water, months it can hang out there. The biggest positive was it was a strong immune response, and you could get 3 doses on a sugar cube, right? So no injections. The negative was something called VAP. Vaccine associated paralytic poliomyelitis. So the disease that polio causes is normally polio replicates in your gut. Fecal-oral route. Makes sense, right? Disease is caused when the poliovirus infects neurons. So the attenuated virus that was created by Sabin was one that should replicate in the gut, so you get this nice strong immune response, but not infect neurons. But what happened was, in some people, it was, I think the numbers were like 1 in 10 million. It was a really, really low incidence. But in some people, the virus would accumulate mutations and infected neurons and caused paralysis. So in this country, because we also had access to the Salk vaccine, which I'm going to talk about in a second, we stopped giving the oral, so this is also called the oral polio vaccine. We just said we don't care if there's any risk of disease. We don't need that. So at a similar time, Salk was making an inactivated where they actually used formalin, so a type of formaldehyde, to treat the virus so that the proteins were cross-linked and the poliovirus could never uncoat. So the big positive was absolutely no vaccine-associated paralytic polio. But some negatives is that it must be injected. Stop. Not as strong or not as strong, long-lasting immune response. And you also had to have 3 doses, but 3 injections. In 1955, there was a batch of inactivated vaccine that was not completely inactivated. And what happened was it actually gave people polio. So the vaccine caused polio, not due to mutations, because they didn't correctly inactivate it. So as I tell you all these stories about vaccines, remember that most people that are anti-vaxxers are not because they know any of the biology. They're really worried about something we'll talk about in a few minutes. But there are, some kind of horror stories with vaccines. Okay. Let's see. So, polio is one of these viruses that we have been trying to eradicate. So I did my graduate research from, how long did it take me? 1994 to 2000. I think I was in graduate school about 6 years. Yeah, I don't know why I'm trying to count. Okay, so anyways, I worked on polio virus. What we were doing in my lab, we were using, because we knew polio has a nice, safe vaccine, we were actually trying to move HIV genes into the poliovirus genome and create this combination vaccine that would protect you against polio and HIV. Obviously that didn't work because I'm not rich and famous and we have no vaccine for HIV, but that was my work. And while I was working in the lab, we were so afraid that polio was going to be eradicated and they were going to stop our research and we were going to have to start all over. And again, I finished my PhD in 2000, so we're still now in 2019 and still haven't been able to eradicate this virus. And part of the problem is, if you look at countries that have travel alerts for polio, these are countries that are very unstable politically. So lots of warring going on. So it's very hard to get vaccines to these people to protect them. As much as the eradication goal is still working, we're still having a hard time really stopping or eradicating, wiping out poliovirus. Influenza is another virus we'll talk about a couple times, and it's a big vaccine one, right? Because every year they say, okay, the flu vaccine is here. Come get your shots. Well, what happens with influenza is it mutates rapidly. So, in February of each year, they make predictions for what strain of the flu they think is going to hit North America in the fall. And they do this based on, you can see this, disease surveillance and who's getting sick over on the other side of the world and what do we think is going to happen. And so the CDC will work and other labs, commercial labs will work, and they'll look at all these different bits of information about what the outside of the flu virus is going to look like, make a prediction, make a whole bunch of vaccines, and hope for the best shot, right? And I think last year it was like a low, like 30% vaccine efficiency for the flu. So it's hard to predict how the hemagglutinin and neuraminidase, that's the main outside components, are going to change. Influenza also currently is grown in eggs. And so it's actually not as antigenic as the real virus because it's an egg-adapted influenza, right? So the HA and neuraminidase out here have to be able to infect an egg cell and replicate in there. And so the virus is just not, the vaccine for influenza is just not a perfect vaccine, and that's why they create a new one each year. Here's smallpox. Okay, this is just an amazing picture to me. Smallpox was eradicated, officially eradicated from the world in 1980. And the last known natural cause of smallpox was in Somalia in 1977. Here is a boy with smallpox. And here is the same boy with no infection. Quite dramatic. I mean, you can just look at all those pustules and what's happened to this poor kid. He survived, which is wonderful. They were both exposed similarly. Why one person got it and the other, that's always the question about your immune system. So how did we eradicate smallpox? Well, remember we used a related virus called vaccinia. And smallpox was, eradication was successful because vaccinia virus made a long-lasting immune response. There's no animal or environmental reservoir for smallpox. So it doesn't live in an animal, it can't hang out in the environment. That's an issue with polio. Okay, polio is very stable in the soil and water. You can easily identify infection, which means you can quarantine and just try to stop the spread. Right? Polio, you don't know. It's infecting in your gut, and until you get paralysis, you wouldn't know you were infected. The antigens are very stable, so smallpox does not mutate rapidly. And the virus is not persistent or latent. Does not go. So you basically get the infection, your body fights it off or you die, and you're done. You clear it, okay? A persistent infection is like HIV, where you're infected for life, okay? You're always making a little bit of virus. Latent virus infections are like a herpes virus infection. So if you get cold sores on your mouth, and they pop out every once in a while and then they go back, that's called a latent infection, where the virus is kind of hanging out, kind of hibernating for a while, and then gets stimulated to cause an infection, and then goes dormant again, back and forth. So luckily smallpox does not do this. So there's no hidden place that smallpox is hanging out, and that's why we were able to eradicate it. Chickenpox and shingles. So this is caused by, both diseases are caused by the same virus called varicella zoster. And if you look back, we get a chickenpox vaccine now, and it's an attenuated live virus. So chickenpox, varicella virus, goes latent. So it's very similar. It's actually in the same herpes virus family, and it hangs out in neurons. And usually doesn't cause anything. So usually you get chickenpox, you get over it, nothing's going on. But as you get older, it can get reactivated. So chickenpox causes these red dots, not so much bad as smallpox, but itchy and scabby and all of that. Shingles is the name of the disease when chickenpox is reactivated. I should have put some pictures, but you get this very specific kind of rash in an area, a lot of times on the side of your body or your back, and I've heard it's very uncomfortable, very painful, and kind of the worst outcome is it can cause long-term neuropathy, which is just this aggravation of some of your nerves. So one thing I want to point out is that just because you got the chickenpox vaccine doesn't mean you're prevented from shingles. So I thought this was really interesting. Because they're giving you attenuated live virus, it can go latent in your cells and it can reactivate and cause shingles later. My assumption is that they cannot get an inactivated chickenpox vaccine that works well enough for long-term protection. And come on, pen. That's why they have to use this attenuated live virus. So as you get age 60 plus, they start recommending you get a shingles vaccine. And there's a couple different shingles vaccines out there. There's Shingrix, which is a subunit vaccine, so no chance of causing disease. This is great for older people, immune compromised, absolutely no risk of infection. There is also Zostavax, which is basically a big dose. Is it a big dose or a smaller dose? Oh darn it, no I can't. But it's basically the chickenpox vaccine, so it's an attenuated virus. So my understanding is that they are now recommending the Shingrix subunit vaccine because it's actually very good at stimulating the immune response. There's no chance of disease. It's better for preventing post-herpetic neuralgia, PHN. Post-herpetic is talking about the herpes virus, that this is in that family. And neuralgia, I was talking about this constant pain due to kind of inflammation of neurons. Oops. So, Shingrix is now kind of the new recommended vaccine, but they're still giving out Zostavax, and so some people report different side effects with each of these. Luckily, I think y'all are way under 60, so you don't have to worry about it. I may sound old, but I still have a few years to that point too. Okay, so just to understand too that chickenpox and shingles, two different diseases caused by the same virus, varicella zoster. All right, backlash from Andrew Wakefield. So this is where the anti-vaxxer thing started coming. This guy produced a scientific paper that linked autism to vaccines. He falsified data. He was charged with misbehavior to developmentally challenged children. He pulled the data that he wanted that supported his hypothesis of the cause of autism and published that. And as you can see, we started to have an increase in measles and in mumps, because we are starting to get a decrease in vaccinations. So you've heard in the news, we've had outbreaks of measles worldwide. One of my students presented on the measles and talked about actually in Poland, the government requires you to get vaccinations or you're charged thousands of dollars in fines, and people still are protesting. So I don't know, this is going to be something interesting to watch, to see what happens with anti-vaccination movements and disease occurrence. So, summing it up, some of the challenges with vaccines is how to deal with an ever-changing virus. Okay, so like the flu, constantly mutating. How do we deal with that? And we have a paper that talks about some of the strategies to try to make it more universal. How do we distribute vaccines to war-torn countries? Right? How can we protect people that don't need to die from some of these preventable diseases if we can't get the vaccines to them? And what goes along with that, not just in this country, but in other parts of the world, is how to overcome fears of vaccines. So it's not that people all over the world want these vaccinations and they don't have access to them. Some people don't understand vaccinations. Some people are against vaccinations. Some people in different countries think that the U.S. is trying to use them as guinea pigs, or the U.S. is trying to give them a disease, or, you know, there's a lot of fear of another country saying, hey, we've got this medicine, you need to take it. So there are lots of challenges still with vaccines and developing them. One of the things that I was hoping would come, and they're still working on, is they call it edible vaccines. So this would really be like subunit type vaccines. Wouldn't it be cool if we could grow a vaccine in a banana and you eat the banana instead of getting a shot and you're vaccinated? One of the biggest, it's not hard to express proteins in different food sources. We have genetically modified food now, but how do you control the dose? So how do you make sure this banana produces the right dosage and not too much vaccine or not enough vaccine? Kind of one of the newer ideas with vaccines in plants is maybe you grow it up in this plant and then you dry it down and maybe you make it into a little pill or a little powder or something like that you can control the amount of vaccines that are given. So anyways, lots of research going on into vaccines. I have 2 more topics I want to talk to you about. I know this is a long lecture, so feel free to pause at any time. Immunotherapy or passive immunity. So I've been mentioning antibodies, really important for preventing infection. So the idea here is that you provide a person with pre-made antibodies. And this is going to make immediate protection. So you don't have to wait for the B cells to get stimulated and start producing antibodies. You get immediate protection. The negative is that this is very short-lived. You are not stimulating the immune system, so it lasts about 30 days. So there's no long-lasting effect. But this is great for immunocompromised people, infants, elderly, transplant. See, I can't spell and talk. Compromised people. This is why breastfeeding is so important. In the first 6 months, the immune system is very slow at getting going in a baby, and so the mother is providing antibodies in her breast milk to protect the baby from infections. This is why it is encouraged to breastfeed if you can. This is why in other countries we don't want to tell mothers not to breastfeed. Even if we talked about in transmission, there's a potential for virus transmission in the breast milk because there's so many benefits to breast milk besides the protein and the fluids. You've also got antibodies. Another potential issue is that you can make, some people will make, antibodies to the antibodies, right? So you may not be able to use immunotherapy every month because you may have made an immune response to the immunotherapy. But this is one of the options. Immunoglobulin, if you've heard of that, they're talking about antibodies. And so sometimes with the, if you're exposed to rabies, they will give you a shot of anti-rabies antibodies and immunoglobulin to try to help prevent that virus from moving to the site of infection into your brain and killing you. All right, so that's immunotherapy. Immuno always means antibody. Final topic, a couple slides. Antivirals. Stopping the viral infection. So we're not preventing it. You've already been infected. You're starting to mount immune response. So what can we do? And you're going to find that there's not very many antivirals out there. We have tons of antibiotics, right, to stop a bacterial infection. But the challenge with a virus is you have to find something specific to the virus, right? Because remember, these viruses are taking over our cells. So you can't just kill the cells that are infected because you're going to kill off your host cells. So you have to target a virus-specific protein. And yes, there are lots of them, but it's not easy to make medicines that target these to get inside those cells and to work in the right time frame. Another big issue is mutations. So just like antibiotic resistance, viruses can become resistant to antivirals. So here's just a few examples of different places that we have antivirals to block different parts. So we have some, I'll show you the next slide, some antivirals for the flu that will block the uncoating. We have viruses that block nucleic acid synthesis, so a lot for HIV. But there's also some for herpes. So acyclovir is very common if you have lots of cold sore or herpes virus breakouts. Protease inhibitors for HIV. Virus release inhibitors for influenza. Trying to block viral attachment. So really a prevention. And again, the challenge of the timeline of virus infection and how infected you are by the time you show symptoms. Again, we don't really have a lot of antivirals. And for a virus like HIV, because it mutates so rapidly, we have to use multiple combinations of antivirals to try to overcome this issue of mutations leading to resistance. So here's just your final slide for this, right? Continue recording, sorry. Final slide. Just wanted to show you influenza. Here are the old uncoating blockers. So they block uncoating of the virus so that you don't get infection. Here is Tamiflu. Oh, go away. And Relenza. Oops, I have them opposite. Tamiflu is Ostelamir, Relenza is Zanzimir. What they're doing is, I mentioned in the virus replication cycle, influenza has to go through a maturation process. And so they actually block neuraminidase so that it can't go through maturation and the influenza virus can't be released from the cell, so it doesn't go on for future infections. Again, because of timing, if you don't start Tamiflu or Relenza within 48 hours of a virus infection, a flu infection, it's too late. It's not going to do any good. And all it really is doing is hopefully shortening your infection time. Okay, that's what I got for you for vaccines and antivirals. Thanks.